Okay, I first would like to make an announcement and clarification of a mistake I made. Previously, I said the examination, the final examination is going to be May 10th, which is wrong. Okay, I got that information probably from another UH. So our final is still going to be May 7th, which is a Friday. The time is going to be class time from 11 to 2 o'clock. Still May 7th, about 17 days from now. Okay, so I'm going to, so if you check my syllabus, I updated that information. I'm going to announce this again and again so everybody knows the correct exam time. At the end, in the very last lecture, I'm going to point out some of the key concepts and the key lectures that you need to focus on. I wouldn't say that I don't have problems from other lectures, but if you focus on the lectures and the concept I emphasize, you'll be able to answer the majority, I would say 90% plus of the questions that I will have. So do you have questions about my final exam?
Oh, Professor, did you say it was from 11 to 2 o'clock, so it would still be 3 hours?
Yes, still going to be 3 hours.
Okay, thank you.
Yeah, somehow when I check a UH final schedule, I just want to double check, and then UH Victoria or somewhere that jump out. They just happen to have the same schedule, the Monday, Wednesday, Friday class, and I picked it up.
I do have one quick question, Professor. Do you think rewatching your recorded lectures would be a good way to, and like taking notes on those lectures would be a good way to review for your final?
Yes, I think so. Okay. Yeah. I would say the questions are not going to be very hard. So if you sit in the class all the time, you have a good chance to answer 90% of the questions.
Nice.
Yeah, it's going to be very simple but conceptual. I'm not going to go to the details of terms, but something like what's the 4 factors of Yamanaka factors.
I see.
Yeah, that type of obvious question, you absolutely will have to, get that.
Right.
Like another example, like if I name Irving Westman, you should know that's a person who worked in hematopoietic stem cell.
Okay.
Yeah, that type of simple, very, very simple questions.
It's not bad.
Okay, yeah. So I'm going to start today's lecture. I think I have started to record. Let me just double check if I'm recording. Where is it? Yes, I'm recording. So today we're going to continue to discuss the application of stem cells. And today's content actually is very important because we're going to discuss how stem cells age and the concept of cancer stem cells. Although it is still a debatable concept, cancer stem cells does have provide a lot of insight into designing therapeutic approaches. So that's a very important lecture today. So we'll start with aging of stem cells. As people age, just like many cells in our body, stem cells age as well. So the aging of stem cell probably is one of the most important reasons of aging. So in the future, if we understand the mechanisms of how stem cells age, maybe we can extend the lifespan of human beings by a lot. Because as you can see, a lot of people start to experience problems at elder age. It's because the organs start to, I wouldn't say fail, but the organs are not working properly or as good as when they were younger. It's because the repairing function of stem cells are dropping. for some of the organs that heavily rely on that self-replacement, like in the small intestine and hematopoietic system, as well as in muscle, you can predict, right? The digestion system are not going to be as good, and the ability of the hematopoietic system cell to carry gas and nutrient and also to fight diseases, right, are dropping because of the lack of functional stem cells. And of course, their muscle mass are dropping, function is dropping, and eventually the activity level is much lower. All will contribute to aging, and eventually most of the organs start to suffer, and eventually, people die. So on this slide, I'm going to describe the deterioration of the molecular control that cause disastrous consequences in stem cells. So look at the picture, the illustration there. In young stem cells, what you're going to see is what we described, the criteria for stem cell. They can self-renew and they can generate the lineage cells or terminally differentiated cells very properly. In the small intestine, the crypt is going to generate a lot of cells, not only the epithelial cell, but also the endocrine cell, the immune cell, all kinds of cells that are required to perform a proper function. So that's what happens in a younger individual. But when people get older, the stem cells, especially the adult stem cells, start to have some problems. On this slide, we describe 4 different situations. In situation B, here, what you can see is, can somebody please mute yourself, because I can hear a lot of noises there. Okay, so in situation B, over time, the stem cells lose the ability to go into differentiation. That sometimes happen not to all the lineages, not to all the not to all the terminally differentiated cells. Sometimes it only affects some destinations. For example, like a hematopoietic stem cell. Over time, you still have the same amount of stem cell, but those stem cells have difficulty generating lymphoid cells. But they're still able to generate the so-called myeloid cells. They just have problem generating lymphoid cells. So that is a problem of lineage specification. Although on the surface you see that you still have the same amount of stem cell, the cells still differentiate, but somehow because of a clonal expansion or loss of certain clones, you remember when we describe adult stem cells, sometimes we mention the cells have bias, okay? So some cells prefer to go into one lineage while the other cells prefer to go to other lineages. And now, when the cells age, some of those clones will get lost. Therefore, you start to have a bias towards certain lineages. That will cause problem. For the hematopoietic system, you can predict if you cannot generate a lot of lymphoid cells, that's T cell, B cell, natural killer cell. Those are important, functional cell in our system to fight diseases. And now, the immune function will not be as good. So that is the first mechanism. When adult stem cells get old, they lose the full range of lineage specification. And in situation C, what happens is the stem cells cannot self-renew themselves. So each cell division will generate a progenitor, and then the progenitor will just push down toward the terminally differentiated cell. So you can predict over time the pool of stem cell will get smaller and smaller. Apparently, that's going to be a problem. Eventually, it gets exhausted, and you don't have any more stem cells to use for further cell replacement. So that's apparently a problem there. And in situation D, what happens is the cells can still self-renew. The stem cells can still self-renew, but some of the cells gradually go through a process called senescence. Senescence just means cell aging. The cells will become older, the telomere get shorter, and then eventually they cannot divide any longer. If they cannot divide, of course, they cannot self-renew any longer. So over time, the stem cells can get old. The term is called senescence. So if you compare situation C and D, they're quite similar. They both involve the loss of stem cells just by different mechanisms. One's because they cannot self-renew. The other is the cells gradually will lose the ability to self-renew. So but situation C and D are quite similar. And eventually, because of that, you don't have the ability to renew the cell loss in the organ. And in situation E, what happens is to the opposite of situation C and D, what you see is increased self-renewal. So somehow, maybe because of mutations, the cells now become more and more active in proliferate, and they just self-renew a lot. They replicate a lot, but those cells have problem getting differentiated. And you can predict if that's the situation there, it's likely to cause a tumor in that organ because the cells cannot differentiate, they just expand and expand, that will grow a big tumor. So that's maybe one of the theories about carcinogenesis. Not every tumor is from this type of mechanism, but some tumor may be from the uncontrolled proliferation of stem cells, adult stem cells. Okay, so with aging, we describe 4 different situations. That's what will happen when the stem cells will get older. Okay, on this slide, I showed a table of what will happen for adult stem cells in different systems. Let's start with hematopoietic stem cell. You will be surprised that with the course of aging, actually the amount of stem cells will increase. You will have more adult stem cells in the bone marrow. The number will increase. And there will be some very special phenotypic characteristics. That is what I described previously. You're going to have increased myeloid commitment. So you have increased the myeloid, but you have a decrease in lymphoid commitment. So the lymphocyte, T, B, natural killer, those cells will have less numbers. So although they are still able to differentiate it, but you have a bias toward myeloid and you have less lymphoid. So in the intestinal stem cell, you also will observe an increase with aging, an increase of stem cell number with aging. And what you observe is increase the villus height. So the villus is the structure we described. That's the protrusion part in the small intestine into the small intestine lumen, right? And then the height of the villus will increase. That means greater cell numbers. That may be the underlying reason you see an increase in stem cell number, because now the height of the villus is higher. That means you will need a lot of cells, more cells, right? That probably is the reason why you have an increase in cell number, stem cell number. And you also have increased the Paneth cell number. Paneth cell is the niche cell that provides signals and maybe some of the nutrients to the stem cell in the crypts. Okay, but although we have this observation, you have increased the villus height, increased Paneth cell number, but the significance is not well understood. So we don't know whether it is because of intrinsic mechanisms or extrinsic mechanisms. We don't have much insight for intestinal stem cells. In skeletal stem cell, which is so-called satellite cell, you're going to see a decrease with aging, decrease in cell number, in stem cell number with aging. And as a consequence, there will be a decrease in myogenesis. That's why, possibly why, the older people will have less muscle mass. That could be a reason. But there will also have a phenotypic switch. Now you have an increase in fibrogenesis and adipogenesis. So that means connective tissue will start to grow inside the muscle. And also you're going to have adipocyte. That's fat that will occupy some of the space within the muscle. So that the muscle will become fatter. The muscle is not, with that, you can tell the muscle is not going to be as powerful any longer because you have other tissues growing within the muscle fiber. For neuronal stem cells, also there will be a decrease, and you see a decrease in neurogenesis, but there will be an increase in gliogenesis. See, almost 3 of the 4 most important adult stem cells, no matter you see an increase in stem cell number or decrease in stem cell number, the common thing is there is a phenotypic switch with aging. So usually it means some of the important lineages start to suffer, and other lineages start to dominate at the cost of the loss of the important lineages, as we'll see there. So if we talk about the mechanism of why, you see this kind of change, sometimes the reason is described as intrinsic. Intrinsic means the mechanism is within the stem cells. Some mechanisms is described as extrinsic. Extrinsic means that the reason for the loss of stem cell function is not because of the stem cell itself, but the niche cell, the environment. We use that 2 terms to describe, the reason or the mechanism of adult stem cell aging, and then how you will prove that for one stem cell type it is because of intrinsic reason, or for other stem cell type it's because extrinsic reason. On this slide, I tell you the experiment design. The principle here is called heterochronic transplantation. So basically, you isolated the stem cell either from old or young adult, and then you do the transplantation into either old or young recipient. Okay, so of course you can do different combinations, and this type of experiment is called heterochronic transplantation. So whenever you take a stem cell, you put into the recipient of a different age, that's called a heterochronic transplantation, and that will show you whether that the mechanism of aging, stem cell aging, is intrinsic or it is extrinsic. Let's study 2 examples here. So the first example is about muscle stem cell, the satellite cell, right? And now you can isolate the satellite cell. It's also called muscle stem cell there. And here, the young age is indicated by a pink color, while the old age is indicated by blue color. As you can see, the cells are now from 2 different sources. In one, it is from young animal, while the other is from old. Okay, but now when they transplant the satellite cell into recipient, the recipient will also have a different age, right? So again, the pink color now in a box, right? In a square-shaped box there is the pink indicates young age, while the blue indicate old age. So as you can see, if you transplant a young stem cell into a young niche, a young recipient, what happens is, of course, regeneration, right? But if you transplant an old stem cell into a young individual, you still see regeneration. So this, as indicated by this plus sign. So that means the niche is more important. Okay, you inject either the young stem cell or old stem cell, it doesn't matter. As long as the recipient is young, regeneration will occur. And now let's check the opposite. From here, you can see if the recipient is old, and no matter you inject a young stem cell, young satellite cell or old stem cell, you don't have regeneration. So this, from the other side, again confirmed that the niche is more important. Therefore, you can say that the aging of satellite cells is mainly because of extrinsic mechanisms. Okay, it's how you do the experiment and then draw a conclusion. It's mainly the niche that decides the outcome of stem cell aging. And let's observe what will happen for hematopoietic stem cell. And this is, I shouldn't say this is hematopoietic because what they isolated is the stroma stem cell. Stroma stem cell is mainly mesenchymal stem cell. Okay, in this case, or shall I say it's a mixture, all the stem cells in the bone marrow. So you take the bone marrow, you isolate the hematopoietic stem cell, and then this can be from young or old individuals. That's the donor, and then you do the same experiment. You put into young or old recipient, and now let's observe the result. In this case, if you transplant the young stem cell, you have regeneration, but if you transplant the old stem cell, you don't have regeneration. Although, the injection or the transplantation is into young individual, young recipient, it doesn't matter. And that's confirmed from the other side. And now you inject either young or old stem cell into an old recipient, and the same thing can happen again. If you inject a young hematopoietic stem cell, you have regeneration. Old one won't have a regeneration in old recipients. So from these two experiments, these two heterochronic transplantation of the bone marrow cells, what you can tell is the stem cell itself is more important. Okay, so you can conclude that the aging of hematopoietic stem cell is mainly because intrinsic mechanisms. Okay, so this is somewhere I want to tell you again. This is, if I'm going to test you in the exam, this is the same I would test you. So you don't have to memorize a lot of things, but you have to understand, what is an intrinsic and what is an extrinsic mechanism in terms of aging. So you can predict if there's some isolated concept I mentioned only once throughout the course of this semester, most likely I won't test it. But if there's something that's interesting, that have some thinking involved, or we talk about a term many times, usually I will test it. Okay, on this slide, we're going to discuss why stem cells will age. Basically, we're going to describe the two types of situations, because the adult stem cells usually will have two types of existence. In the first occasion, they could be quiescent for most of the time. But for most of the time, except the hematopoietic stem cell and the intestinal stem cell, they are probably very active a lot of times. Most of the adult stem cell are spending most of their time in quiescent status. That means they sit there, they don't proliferate, they're waiting for their time. If there's injury or the situation calls for proliferation, they can go back to cell cycle. But no, most times they're quiescent. They're in G0. They just, they don't perform any function. They just maintain their, their status. That's their main task. So most times they're quiescent for most stem cells. And in these cells, there won't be a lot of reactive oxygen species strand generation, ROS generation, because those cells have low metabolic activity, right? Because they don't perform particular function, they don't proliferate, they just maintain the status. So they don't have a lot of, mitochondria in there. The ROS generation is very small. And also in this type of cells, they have a better antioxidant system. So that will also contribute to lower ROS-induced damage, because the ROS can induce DNA damage. That's a cost of living, the cost of activity. When we have a lot of activity, your mitochondria is very active. At the same time, you're going to generate a lot of ROS. The ROS is oxidizing agent. It can cause damage in DNA and in some other proteins and organelles as well. But our system have a lot of reductant that can fight that. The reductant are called antioxidant. Okay, so in these quiescent cells, they have a very good antioxidant system that can be used to control the damage by ROS as well. Okay, so you won't see a lot of DNA damage here for the quiescent cell, but the problem is if damage does occur, because although it is kind of rare, but it can still occur. The problem in a quiescent cell is if there will be damage, the damage will be repaired mainly by non-homologous end joining. So if some students are interested in CRISPR, Cas9, maybe you're familiar with this term, non-homologous end joining, NHEJ. NHEJ is an error-prone mechanism in repairing DNA. Basically, when they link this double-strand break together, they cannot link it perfectly. So they will insert some nucleotide or remove some additional nucleotide, therefore that will cause a mutation there. So that repairing is not perfect. It will cause mutation, therefore it will contribute to aging, contribute to stem cell aging. But the stem cells, like the hematopoietic stem cell and intestinal stem cell, and most of the other adult stem cell, they will have to proliferate at some point, right? So they will proliferate although most of the time they're in quiescence, but throughout their lifespan, they will have to proliferate a few times. And when the situation calls, they will have to proliferate and then generate the lineage cells. So proliferation does occur for most stem cells. So for the cycling stem cells, they face other problems. Although in these cells, if you have a DNA damage, the good thing is the damage are usually repaired by homologous recombination. Homologous recombination means the cell will use a template to do the repair. For example, you have 2 copies of DNA, so they can use the other copy as the template, therefore do a perfect job, close to perfect job to get that repaired, the DNA damage repaired. When they have a template, they can repair just based on the template. Therefore, it's almost perfect repairing there. But in these cells, because of their cycling, they have a lot of protein translation, transcription, and also the metabolic rate is much higher. You're going to see a lot of mitochondria in the cycling cell, and therefore there will be a lot of ROS generated. And because of that reason, this more ROS will not have enough antioxidant to balance it out. Therefore, more DNA damage will occur. So although you have a very good repairing system, but there will be a lot more damage caused by the increased amount of ROS, and eventually that will also cause DNA mutation and aging of stem cells. So here we describe two situations, one in quiescent cell, the other is in cycling stem cell. They have different mechanisms in there, but both will experience aging because of the properties of them, the intrinsic properties of them that cause them to be vulnerable to aging. More mutations than most other cells. So in the textbook, it's a long chapter. You can read this through and then understand the additional aspect of stem cell aging. Since today we're going to discuss cancer stem cell as well, so I'm going to just describe another very important aspect. That is metabolic activity. How metabolic activity is related to stem cell aging. So in this illustration here, what the textbook describe is that the metabolic activity, the metabolite, contribute a lot into to epigenetics. How DNAs and histones will be modified will be dependent or decided by metabolic activity. Why? It is because two of the most important, very important pathways in metabolism contribute to or is directly linked to DNA modification and histone modification. For example, like glucose metabolism will produce alpha-ketoglutarate. Alpha-ketoglutarate is one of the 8 intermediates in the TCA cycle, right? I think a lot of you guys are from biochemistry. Alpha-ketoglutarate is one of the very important intermediates in the TCA cycle. But alpha-ketoglutarate is a cofactor for two very important methylases. One is TET. It's a DNA demethylase that removes the methyl group. Okay, so demethylase, it removes the methyl group. But other is JMD. The JMD is a so-called Jumanji family of demethylase. JMD works for histones. It's for histone demethylation. So as you can see, immediately the glucose metabolism is related to DNA and the histone demethylation. So they provide an important cofactor. And glucose metabolism, of course, will also produce NAD+, right? They use NAD+, and then through the TCA cycle and the oxidative phosphorylation, as well as the 11th step of glycolysis, you have tons of mechanisms that will convert between NAD+ and NADH, right? So you always have to have a lot of NAD+ in our body. That's the oxidizing power there. But the amount of NAD+ is also important because it's a cofactor for sirtuins. The sirtuins is deacetylase. It's deacetylase for histones. So NAD+ is a cofactor that is used for histone deacetylation. So as you can see, the glucose mechanism metabolism is directly related to histone and DNA methylation and deacetylation. So acetylation and methylation are the two most important mechanisms of epigenetic control. So both are directly related to metabolism. And another important metabolite is from 1-carbon metabolism. You learn this not from the introduction in biochemistry, but the advanced biochemistry. So one of the product intermediates in this process is so-called SAM, S-adenyl methyltransferase. Okay, so this is an important intermediate for the 1-carbon metabolism, and this is a cofactor for DNA methyltransferase. For DNA methyltransferase, therefore, the intermediate here is used for DNA and histone methylation. But of course, I cannot say that except histone acetylation. We know histone acetylation is always an indicator of active transcription, right? So the gene will be actively transcribed if you have a histone acetylation. But for other modifications such as DNA methylation, histone methylation, it depends on the situation. Sometimes the methylation will be an indicator for active transcription. Sometimes it's an indicator of transcription suppression. So we can't draw general conclusions, but what you can say is, okay, the metabolism is really very important for histone and DNA modification. Therefore, they're important for epigenetic changes. No matter it's aging or other processes we have in our everyday life, it's always about epigenetic changes, how the DNA is, how the histones will be modified. It's not about mutations. It's not about you lose DNA or you gain DNA. 99% of the time, it's not because of those, but because of modification on DNA and histones. Okay. So although we couldn't draw a straightforward conclusion how metabolism will affect DNA modification, but we do have some general principles we learned over time. That is, when the individual have poor nutrition, aging is slowed. When the individual have or the cells have rich nutrition, aging will be accelerated. So this is very, very important because this is not only important for stem cells, but for us as well, for people, as an individual as well. Because if you experience poor nutrient, what will happen is low insulin signaling, and that will activate pathways such as the FOXO pathway and sirtuin pathway. And next, what will happen is increased autophagy, increased self-renewal, DNA repair, and quiescence. So these are considered to be good. But if the nutrient is rich, what will happen is high insulin signaling, and of course you have a high amino acid and high fat diet, and mTOR will be activated. The mTOR pathway will be activated. Cell will start to proliferate. And start to build up, like a increased translation. That will contribute to aging, contribute to stem cell aging. The best learned example is for satellite cells. So if you have always in a nutrient-rich environment, and eventually you accelerate aging. So there are many papers not describing from the angle of stem cells, purely describing how nutrient will affect lifespan, will affect the overall health of human being. So what they learned is, if you do fasting, at least 1 meal a day, you're going to live longer. That being described many times in the top journals such as Science and Nature. And what they did is very large scale that often involve like thousands of people. And then the conclusion, in my opinion, is very solid. So basically, you intentionally put yourself in a nutrition poor situation. Actually, you're slowing down stem cell aging, and you're slowing down the aging of yourself. Okay, so we just have too much to eat, and it's very hard to resist the great foods.
Yeah, that's a really interesting finding.
Yeah, it's solid science. It's very solid science. You search internet, you will see a lot of papers published in some of the best journals, all pointing toward eat less and skip meals, and then that helps you live longer.
I honestly thought the opposite, but wow, that's a really good
Yeah, I guess that's related to modern society, modern lifestyle.
Yeah.
Yeah, in the old days, of course, when you don't have a lot of nutrients, eat more probably is better. But
Yeah.
now it's very different.
Sure.
Okay, so lastly, I'm going to spend about 10 to 15 minutes describing tumor cancer stem cell. It's a pity we don't have a lot of time to describe this because it's a super important field, newly developed field, is still developing. So tumors, just like an organ, like a liver, like a lung, like most of the organs of our body, the tumor will have a lot of cell types within it. It's never homogeneous. Always have a lot of different cell types. So in tumors, people feel like maybe there is a hierarchy. Just like in most organs, you have a hierarchy of cells. That was later on, I wouldn't use the word to prove, but demonstrated to be somewhat true. So here I would like to describe the basic theory of cancer stem cell. So basically, from a primary tumor of several cancer types, such as leukemia, breast cancer, lung cancer, colon cancer, and osteosarcoma, see, in at least a few cancer types, not in all the cancer types, just in a few of them, people realize that it's to start off, you have many cell types there. So some of the cells, or shall I say, most of the cells are non-tumorigenic. So they're not actually tumor cells. They just happen to be in that tumor, providing some roles, usually a supporting role for the tumor cell, for the true tumor cell. So these cells are also sensitive to chemotherapy. So when you give a chemotherapy, you make the cancer mass to become much smaller when you compare to the beginning. But what you have killed are mostly non-cancer cells. It is very difficult to use terms. I shouldn't say non-cancer cells because they're all tumor cells. Because they are isolated from tumor, they're all tumor cells. But they're not cancer stem cells. They're non-cancer stem cells. They are non-tumorigenic. That means if you take just several of these cells, you put into another individual, another animal, it won't grow tumor. Okay, it's non-tumorigenic. So they're sensitive to chemotherapy. But there are rare populations in the cancer termed the cancer stem cells. They can self-renew, they can proliferate, and they are tumorigenic. So you just take some of those cells, you put into another individual, it's going to grow up a tumor. And in that new tumor, you're going to find all the cells in the original tumor because the cancer stem cells can differentiate and then give rise to the rest cell types within that tumor. So if you try to fit the criteria of stem cells for cancer stem cells, it fits because they self-renew, they also differentiate, they give rise to the other cell types within a tumor, right? You can say that them as cancer stem cells. And a very important feature of cancer stem cells is they're often resistant to chemotherapy. As I said, chemotherapy, the patient will experience a lot and will have to take on the very bad side effects, such as losing hair, but eventually the tumor is likely to come back again because the cancer stem cells are not destroyed. Only the other tumor cells are destroyed. Therefore, the cancer stem cell will take time and then grow up, self-renew, proliferate, and give rise to other cells within the tumor and grow right back. So that's why tumor will recur a lot of times. So on this slide, what I'm going to tell you is an experiment done by people doing breast cancer research. Cancer stem cells was first described by John Dick in, I believe, acute myeloid leukemia, AML. That's when it was first described. But on this slide, was shown is a experiment using breast cancer cells. So basically, they take a tumor from a patient, okay, and then they make a single cell suspension, and then they start to sort the cells into different fractions based on the expression of surface markers. So what's shown here for the first row here is unsorted cell. So you basically, that's a mixture of all the cells within that tumor. You can see they injected a lot for a tumor formation experiment called a xenograft experiment. Basically, you put these cells into an experimental animal, into a lab animal. So if you put the unsorted cell, if you put a lot, like here it's 500,000, you're going to have 8 out of 8 times tumor formation. So if you inject less, like 5 times 10 to the power of 4, still 10 out of 10 times you have tumor formation. But if you further drop the amount of a cell, and now you can see when you drop it to 2 times 10 to the power of 4, you don't have tumor formation any longer. And then you drop even less, essentially you have the same observation. So you don't have tumor formation any longer. So you have to inject a lot of tumor cells if you don't do cell sorting. And now let's see, if you isolate based on surface marker CD44 positive and CD24 positive throughout, you just don't have tumor formation, right? No matter high or low cell number you inject, you just don't have it. That means these cells, they are non-tumorigenic. But if you sort by CD44 plus but CD24 negative or low, what you can see is I'm sorry, here, when they have this slash, it means undone. They didn't do the high number injection, but when they their first start point is 2 times 10 to the power 4, is that you have 10 out of 10 times tumor formation. Even if you drop this to only 1,000 cells, you still have 10 out of 10 times tumor formation. So these cells are kind of powerful in generating tumors when you compare to the CD24 positive. And now they further refined the population by introducing another surface marker called epithelial surface antigen, ESA. The ESA plus cells on top of CD24 negativity, as you can see, even if you drop to 200 cells only, it still can generate tumor. So that means these markers, CD44 positive, CD24 negative, and ESA positive, means those cells are true stem cells, because only small amount of them will be able to generate cancer, while large amount of the non-tumorigenic cell, the CD24 positive cell, won't be able to do that. From this, you realize there is a hierarchy there. The cells, have a hierarchy. So here, I just want to show you the markers used nowadays for different types of cancers, solid cancers. The markers used to describe or identify cancer stem cells. I won't go through this individually, but I just want to point out they have some common ones, such as ABCG2. I mentioned this many, many times. ABCG2 is a channel protein that is related to the site population phenomena. Okay. You see ABCG2 again used to describe several cancer stem cells, and this ALDH1. I apologize. Previously, I said this is alcohol dehydrogenase. It is not. It's aldehyde dehydrogenase. Okay, this is not related to glycolysis. I mixed up concept, but this enzyme is about the chief function is about liver detoxification for alcohol. It's still about alcohol, but the name actually is aldehyde dehydrogenase. Okay, you see this is a marker for multiple cancer types. And again, CD133, you see it in many places. CD133, also named the prominent, is a surface marker for the adult stem cells as well as cancer stem cells. Okay, so I'll use 2 to 3 minutes to say something about asymmetrical division. we said that stem cells, mostly adult stem cells, will rely on asymmetrical division to maintain themselves as well as going through differentiation, right? Because only asymmetrical division will give rise to one stem cell and one progenitor cell, right? So you will absolutely need asymmetrical division for adult stem cell, because if it's not asymmetric, you either end up with two stem cell or two differentiated cell. If it is two stem cell, it is okay for some situation, right? You just maintain it. But if you will only have two progenitor cell, you're going to exhaust the pool, right? So the asymmetrical division is super important. But the mechanism of this has only been described recently, starting from work in Drosophila. In Drosophila, this asymmetrical division also occurs, and people start to realize the mechanism as shown from this illustration. It turned out there is a critical enzyme called apical protein kinase C. It's called apical only because the location of this enzyme, this PKC, is located in the apical region of the cell. Well, the APKC will be able to phosphorylate some of the important differentiation proteins, differentiation-related proteins such as NAM and MIRA, especially NAM. So when NAM is phosphorylated, NAM will have a redistribution. NAM will go to the so-called the basal region of the cell. So as you can see here, the APKC moved to the apical region, while NAM goes to the basal region. Originally, it's evenly distributed. Now it goes to one side of the cell. Therefore, we call this polarization. And then that will cause the spindle orientation based on the apical and the basal region. And that eventually will cause an asymmetrical division. The APKC goes to one cell, while the NAM goes to the other cell. The APKC will be located to the apical region and do asymmetrical division again. So because NAM is now distributed only to the progenitor cell, NAM is differentiation related. NAM will initiate differentiation in that progenitor cell. Well, because in the other cell you don't have NAM, they will keep the stem cell status. It is a very, very smart design, naturally selected design there. That is how asymmetric division will occur. It turned out this has an indication in cancer, because when people did a NAM deletion, NAM genetic deletion using animal models, they observed that the animal will have brain tumors. So that means, the brain stem cells may not be able to go through asymmetric division. Therefore, they can only generate stem cells. They cannot give rise to progenitor cells. The result is tumor formation in the brain. And later on, and then in human beings, it turned out that NAM is downregulated in several tumors, including leukemia, breast, and colon cancer. That is understandable, because when you have a less NAM, this asymmetric division probably will suffer, therefore causing the cells to self-renew. If it is uncontrolled, and it can cause cancer. And to the other hand, you have an RNA binding protein, Musashi, which is an inhibitor of NAM, and this turned out to be upregulated in leukemia, because this is an inhibitor of NAM. Okay, this you can predict that this will also make the asymmetrical division not to function properly. Therefore, the upregulation of Musashi is also found in leukemia. So the asymmetric division have a special indication in cancers. The cancer cells somehow use this as a mechanism, to become cancer. I'll skip this one, the cancer stem cell niche. I hope you will read through this. Basically, in the cancer, although you have cancer stem cell, you also need other cell types. Some of the cells are niche cells. They help the cancer cells to maintain their cancer stem status and also help them to basically there is interaction there. We have 3 examples in brain, in colon, and in skin that describe different niche cells. Why we try to understand the cancer stem cell niche, why we have this concept of cancer stem cell, it is because there's great new possibilities because of this this new concept or new paradigm of cancer stem cell. First of all, as I said at the very beginning, if you use chemotherapy, a lot of times the cancer stem cells will be able to escape. You only kill other tumor cells. Therefore, recurrence is going to be a result of a lot of those therapies. Sometimes you can achieve long-term effect, but sometimes the recurrence can be very quickly. So when we have this cancer stem cell concept, so you can design ways to identify those in vivo, such as imaging. If you can develop some special dyes or special chemicals or special whatever that can be used to mark the cancer stem cell, and then from imaging system, for example, X-ray or MRI, just something, we're able to identify the cancer stem cell. Then you can evaluate how many cancer stem cells are there, right? So although it's cancer, if it's in 2 different individuals, you can use that, the amount or the proportion of cancer stem cells to judge whether, to judge the severity of that cancer. Although it's the same thing. If you do a biopsy, most likely you're going to have some very similar pathological findings. That won't be helpful. But if you can pinpoint to the amount of cancer stem cells, well, you have a better evaluation of the outcome. And of course, if you can mark those cells, you can develop some special treatment just for the cancer stem cell. So you kill the cancer stem cell without worrying about the other cells, because the other cells just become a problem if the tumor grow big. It will put pressure on nerves and on circulation and such. Otherwise, they will be harmless, right? Most of the times. So targeting cancer stem cell is very, very important. But sometimes you want to even target the niche cell. If you know the mechanisms of how cancer stem cell and the niche cell interact with each other, maybe in the future, you don't even target the cancer stem cell. You only targeted the niche cell. So if you cut the interacting between the niche cell and the cancer stem cell may not be able to thrive either, right? So that's the end of today's lecture, and I hope you enjoy it. And we don't have many left. Next week it's going to be our last week, and I want to say something about to group 3. So Friday you're going to have a presentation. It's more complicated than what you had in the first paper that you delivered not long time ago. So if you have question, please ask me, and good luck with your presentation this Friday.
Thank you, Professor, for the lecture.
You're welcome.
It's really interesting because I never thought we would be this close to actually coming up with a solution to get rid of cancer cells for good.